Sushruta for NEET is now available on the Google Play Store. Try it out for free.
I hope you are seeing this board screen. Now let us take up the next part. Vectors for cloning genes in plants and animals. Which vectors are used? Is it that always we have to go for the plasmid present in the bacteria as the vector? You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses. So it is through bacteria and viruses. Yes, it is through bacteria and viruses, and which have been known for ages with regard to the transferring of what we shall see that each have been known this for ages. With bacteria and viruses, they knew about that. You think that what this bacteria can do? Bacteria even it is a prokaryotic cell, but now I am sure you know what viruses can do. Am I right? Viruses have virus has taught us a great lesson. Keep always clean. Is it not? Maintain social distance. Say always namaskara. Don't have shake hand and get any bacteria. So we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacterial viruses want. So bacterial viruses, they used to get whatever they needed by by just delivering the genes into the body of the organism from which they expect something. For example, Agrobacterium tumefaciens. Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as a tDNA. Remember, a bacterium called Agrobacterium tumefaciens, a pathogen of several dicot plants, means a disease-causing organism of several dicot plants, which used to bring a disease in the dicot plants, is able to deliver a piece of DNA known as a tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce chemicals required by the pathogen. I hope you understand this. Jayasri, what is the Agrobacterium tumefaciens used to do? Agrobacterium tumefaciens, which is a known bacterium to cause diseases in dicot plants. It used to deliver a piece of DNA known as a tDNA. T stands for tumor. tDNA to transform normal plants into normal plant to transform normal plant cells into a tumor and direct these tumor cells to produce chemicals required by the pathogen. Is it clear? Is it clear this particular sentence? Nadira? Vismita?
Yes, sir.
Clear, Harshvardhan?
Yes, sir.
So,
Yes, sir.
similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. Can you name any retrovirus? Example? One example?
HIV.
HIV.
HIV is a retrovirus, best example for a retrovirus. A better understanding of the art of delivering genes by pathogens, a better understanding of the art or skill or method of delivering genes by pathogen in their eukaryotic host has generated knowledge to transform these tools of pathogen into useful vector for delivering genes of interest to human. So now, by this bacterium or from this bacterium, we learned that the bacterium can transform a normal cell into a tumor cell and get to the things, whatever it is required, produced by the plants. So this knowledge we are trying to just use for sending whatever we want into different organisms. So a better understanding of this skill method of delivering genes by pathogen In their eukaryotic host, pathogen is a disease-causing organism, parasite. In their eukaryotic host, if the bacterium can deliver some gene, a parasite can deliver some gene to its host, why not we make use of that knowledge? So this has generated. If the bacterium, a parasitic bacterium, can deliver some genes to the host body, we have tried to find out how it is going to deliver, and this knowledge we have used as a tool, whatever that pathogens are used to as a vector to deliver the genes of interest to humans. The tumor inducing, let us call it abbreviated TI, capital TI, tumor, T for tumor, I for inducing, tumor inducing plasmid of Agrobacterium tumefaciens has now been modified into a cloning inducing plasmid inside the body of this bacteria. Tumor inducing plasmid is now modified into a cloning vector which is no more pathogenic to the plants, which are no more pathogenic to plants, but is still able to use the mechanism to deliver genes of our interest into variety of plants. Right? So that particular tumor-inducing plasmid is no more pathogenic to plants, but we are using that tumor-inducing plasmid to deliver genes of our interest into a variety of plants. I hope it is understood. That tumor-inducing plasmid is not used to just induce any disease that, but it is used to deliver genes of our interest into a variety of plants. Is it understood, or any doubt is there? Siddesh, Pratiraj,
Yes, I understand.
Rakesh, have you followed?
Yes, sir.
Yeah. What was the problem with you? Why you called me?
It was asking permission to log.
Okay, okay. You got in. Okay. Understood. So now, Vishwita, have you followed it?
Yes, sir.
Similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. Disarmed means made less pathogenic, non-pathogenic. Retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial or plant or animal host where it multiplies. So whatever gene of interest, whatever the desired gene that is ligated to suitable vector need not be only one particular animal, one particular organism, one particular plasmid. Now we have seen that tumor-inducing plasmid also used here. So once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacteria or a plant or animal host. Now let us see what is done. Now, competent host for transformation with the recombinant DNA. Now the thing is that you got a gene of interest, you got a vector to carry this gene of interest or desired gene, and you are interested to put it inside the body of the host. Now the question is that, is it that easily we can put it into the body of the host? That's a big question. Can we easily insert a plasmid carrying the desired gene, or carrying the desired gene, or a plasmid carrying the gene of interest? Can we easily put it, introduce into the host? That is the question. Since DNA is a hydrophilic molecule, water loving molecule, it cannot pass through cell membrane. Because why it cannot pass through cell membrane? Why DNA cannot pass through cell membrane? Rafa, why
Yes,
DNA
sir.
why DNA cannot pass through cell membrane? Aisha, Blessy, Kanisa,
Yes, sir.
why?
Is it because, sir, it's semi-permeable?
Semi-permeable membrane made up of what? Yeah, made up of. Now, yeah. Anyone else? Nadira, Savitri,
Yes, sir.
Jayasri, Tanusri, Namrata, Jyoti. Hello, Lohit. Why it cannot pass through cell membrane? What is the component of cell membrane? You don't remember? Anyone? Prithvi? Hello?
No, sir.
Siddesh? Harshavardhan? Who else? Charan? Vismita?
Yes, sir.
You don't know?
Sir, is it because cell membrane is made up of lipid?
And protein. Bilayered lipid sandwiched with the protein. And this will not permit the DNA like molecule which is water loving to pass through. Can you pass water through oil?
No, sir.
No. In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. What has to be done? The bacterial cell must first be made competent. How to make competent? That means DNA should pass through the wall of the bacteria. Okay? This is done by heating them with a specific concentration of divalent cation. What we are doing? We are heating these bacterial cells with a specific concentration of divalent cations such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. So, the moment this bacterium is heated with the calcium, treated with the calcium, it makes the pores to get open, bacterium. So this enables the DNA to enter the bacterium through pores in its cell wall. So this is making what? This making the bacterial cell wall competent enough to take up the DNA. Okay? Right. So, now, look here. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA. We are incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees centigrade heat and again putting them back on ice. Ice, heat, ice. Cold shock, heat shock, cold shock. So first what we have done? We have treated with the divalent ion calcium so that the pores in the wall of the bacterial cell opens. Now we are putting such a bacterium and also we are forcing such a bacterial cell to get this recombinant DNA inside by incubating the bacterial cell with the recombinant DNA, both the recombinant DNA plasmid have recombinant DNA, and this bacterial cell, these are treated on ice, followed by brief treatment of 42 degrees centigrade temperature, and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. Are you following me? Are you following this? Aisha? Blessy? Blessy?
Yes, sir.
Yeah. So this is what we have: chill ice. Okay. We are putting both the recombinant DNA and also bacterial cell here. Then again, 42 degrees centigrade hot water bath. So this hot water bath have 42 degrees centigrade. In that hot water bath, you are placing. Again, you are putting back them on ice. Right? So this is not the only way to introduce alien DNA into host cell. In a method known as microinjection, there is another method called microinjection. Recombinant DNA is directly injected into the nucleus of an animal cell. So we have another technique. What is that? Microinjection. Direct transfer of DNA into the cell without a carrier. Without a carrier means without any plasmid is called DNA microinjection. This can only be done for only few cells at a time. This technique is used mainly for large cells such as oocytes, eggs, and cells of early embryos. The DNA can be directly injected into tissues such as the skin, muscle, or internal organs, or it can be injected into the blood. This is what is called DNA microinjection. Okay? Right, direct injection. Now, in another method, suitable for plants, cells are bombarded with high velocity microparticles of gold or tungsten coated with the DNA in a method known as biolistics or gene gun. So you can see here what is done here. It is the cells are coated with gold or tungsten here, and then suitable for plants. It is suitable for plants. Cells are bombarded with high velocity microparticle gold or tungsten. This gold or tungsten, they are coated with DNA, and then they are introduced. And this is what is known as a biolistic or gene gun. Gene gun. Okay, I hope you understand my point here. This is what is done. Just a cartridge carrying the DNA coating inside. So this is a biolistic or gene gun where gold particle or tungsten particles are coated with DNA and then put inside the host organism. So this is what microinjection, biolistic gun or gene gun, DNA particles coated with the gold or tungsten, DNA coated gold particles, right? Then they are introduced into the target plant cell. I hope you are following now. And the last method uses disarmed pathogen vector. Disarmed pathogen means they are not, their power of causing disease is reduced or taken away. And the last method uses the disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Okay, you know, pathogen attacks the host. So this pathogen is without the ability to cause the disease, but it is carrying DNA. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the process of facilitating recombinant DNA technology again. That means whatever you have studied, we are just going through once again. So I am not taking much time for this because all this whatever you are going to learn now, study now, this is a repetition what you have studied in the last 3 classes. Let us see that. Okay, 1 minute. So, processes of recombinant DNA technology. Recombinant DNA technology involves several steps. It involves several steps in a specific sequence, such as you know, isolation of DNA, fragmentation DNA by restriction endonucleases, isolation of the desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at a large scale, and extraction of the desired product. This one paragraph will tell you all the points, all the steps required in a DNA, recombinant DNA technology. Am I right? Kanisa? Am I right?
Yes, sir.
All these we have seen. So now let us examine each of these steps in some detail. Isolation of genetic material. What is that? Isolate genetic material. What is that genetic material we called so far? What is that? What is that?
Desired gene.
Desired gene. Recall that nucleic acid is the genetic material of all organisms without exception. In the majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with the restriction enzymes, it needs to be in a pure form, free from other macromolecules. What other macromolecules are found along with the DNA? Can you tell me? What other you will see along with the DNA? What is there surrounding the DNA? You have looked into an experiment to know which is the genetic material, DNA or
Protein.
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 So you come across it. Since the DNA is enclosed within membranes, we have to break the cell open. Where the DNA is present? DNA is present inside the cell. It is present inside the nucleus. Nuclear membrane is there, and it is present in the chromosome. And you know, chromosome has a basic histones associated to the DNA. So, since the DNA is enclosed within membrane, we have to break the cell open to release the DNA along with other macromolecules what are present in cell, such as RNA, proteins, polysaccharides, and also lipids. So, we must get a pure form of DNA. We must see that this DNA is becoming free of other macromolecules like a protein, the polysaccharides, that is carbohydrates, polysaccharides, lipids, and also RNA like other nucleic acids. Now, how to make a DNA pure? Let us say. Now, this can be achieved by treating the bacterial cell or a plant cell or animal tissue with enzymes such as lysozyme, cellulase, and chitinase. What it does when you treat this bacteria with the lysozyme, lysozyme, it dissolves. Lysozyme are the body which has all type of enzymes to dissolve whatever that bacteria possesses. Cellulase dissolves cellulose. Chitinase dissolves chitin. So in this way, we are using these different enzymes to dissolve all these products that are found around the DNA. You know that genes are located on long molecules of DNA, intertwined with the proteins such as histone. I have told you there are basic proteins surrounding the histone, so they also should be now digested. The RNA can be removed by treatment with the ribonuclease, whereas the proteins can be removed by treatment with the protease. So, water or molecules can be removed by appropriate treatment, and purified DNA ultimately precipitates out after the addition of chilled ethanol. Once you treat this cell, whatever maybe you are taking, whether it's plant cell, fungal cell, bacterial cell, whatever it may be, once you treat with these enzymes, all other macromolecules they get dissolved, they get digested, and you will get a pure DNA. And this pure DNA it precipitates when you when you put it in a chilled ethanol. This can be seen as collection of fine threads in the suspension. So this is what is done. DNA that separates out can be removed by spooling. What you have, this is the DNA. Whenever you just put a spool and rotate, you will get only DNA. You can see the DNA with your naked eye. You can see the DNA. What you have is a DNA, and that is the basic structure of the DNA that you have. Okay? Right? Chemical structure, chemicals. So, cutting of DNA at a specific location. Now we want to get a desired gene. It is not that whole DNA we want. Restriction enzyme digestions are performed by incubating purified DNA molecule with the restriction enzyme at the optimal condition for that specific enzyme. Okay, we are using restriction enzymes. Okay, these restriction enzymes also known as hello,
Molecular scissors.
also known as
Restriction endonuclease.
restriction endonuclease. So these they cut the DNA at a specific what is that? What is that? These restriction endonuclease are in a position to locate, identify recognition sequence
Sequence.
with the palindromic nitrogen bases. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. I hope you understand. Agarose gel electrophoresis. What is done in that? We are cutting the DNA after cutting with the restriction enzyme, the cut pieces are put into the agarose gel so that we see that some of these they run faster towards anode or cathode. DNA is a negatively charged molecule, hence it moves towards the positive electrode or anode. You have seen this, am I right? Okay, so we are putting that DNA with the restriction enzymes into this well. So in this, it gets cut, and these cut small piece of DNA, they run towards anode depending upon their sizes. Smaller the better, faster is the movement. The process is repeated with the vector DNA also. The process is repeated with the vector DNA. What is that vector DNA? Which is vector here? Where is the vector DNA present?
Plasmid.
Plasmid. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with the space are mixed and a ligase is added. Are you understand this point? The cutout gene of interest from source DNA and the cut vector with the space are mixed and a ligase is added. This results in the preparation of recombinant DNA. Savitri, ಅರ್ಥ ಆಯ್ತೇನಮ್ಮ?
Yes, sir.
Okay. Shall we go ahead? Shall we
Okay,
go
sir.
ahead? Namrata? Namrata?
Yes, sir.
Okay. Now, amplification of gene of interest using PCR. This is a new technique. Have you heard about this PCR nowadays? With reference to
COVID.
COVID?
COVID test.
What you have heard?
PCR test, sir. COVID test.
Yeah, what is that? You see it. This PCR stands for polymerase chain reaction. Why it is done? It is just to amplify gene of interest. We want to get more number of gene of interest. With whatever that we get thousands of copies there. This is what we want. So that becomes easy for us to notice whether that particular gene is there or not. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vitro using two sets of a primer. There are two sets of primers are used. Small chemically synthesized oligonucleotides that are complementary to the regions of DNA. Primer is nothing but a group of, a sequence, a chain of few nitrogen bases, oligonucleotides. Okay, and the enzyme DNA polymerase. Remember this reaction, multiple copies of the gene or DNA, gene of interest is a synthesizer. We are making multiple copies because we cannot go for again and again removing that from the organism in which we are interested, and again putting. Once you get a little bit of this gene of interest, we have to be in a position to multiply them. It's a synthesis in vitro. Gene of interest is a synthesis in vitro, in test tube, in petri dish, in vitro, using two sets of primers and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotides provided in the reaction and the genomic DNA as a template. Genomic DNA as template. Okay, so genomic DNA means the DNA that is present in the gene of interest that is present in the original DNA. Now, let us see this one. If the process of replication of DNA is repeated many times, this is repeated. Process of replication of DNA, whatever the gene of interest is there, it gets repeated. You get to multiple copies. The segment of DNA can be amplified to approximately billion times. That is 1 billion copies are made within no time. By this PCR test, it is multiplied billion times. Such repeated amplification is achieved by the use of a thermostable DNA polymerase isolated from a bacterium, Thermus aquaticus. It is isolated from Thermus aquaticus bacterium. Such a repeated application, making one copy into billion copies, is achieved by the use of thermostable DNA polymerase, which remain active during the high temperature induced denaturation of double-stranded DNA. What happens? Which remain active during the high temperature induced denaturation. You have studied earlier that even if at all it is heated, DNA will not get denatured. It will not lose its capacity. So by heating, we can make that the two double-stranded DNA, it can get separated. It can get separated. Okay, but it is stable. So such repeated applications achieved by the use of a thermostable DNA polymerase, then thermostable. That means It is stable at high temperature. Thermostable DNA polymerase, which remain active during the high temperature, induced denaturation of double-strand DNA. Even if at all it is subjected to high temperature, no doubt the two strands of the DNA get separated. It enables us to get multiple copies of this gene of interest, but nothing happens because of temperature. Its quality is not affected. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Are you understanding this point? Have you understood? Have you understood this point? Blessy? Kavita? Kavita, are you there?
Yes, sir.
Have you understood?
Yes, sir.
Blessy? Blessy?
Yes, sir.
Can you go ahead? Janani? Janani?
Yes, sir. Yes, sir.
Okay. And one thing, if at all I don't call your name, please don't think that I have forgotten. Whatever the name it comes immediately, I will utter. And it is not that I give any importance to anyone, less importance to anyone like that. Okay? Okay? If at all I have not called your name, please don't find fault in me. You only start asking some doubt so that I can remember your name very well. Then my question will be to you. Understood? Lohit? Lohit?
Yes, sir.
Diti? 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 Hello? Okay. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Okay? This is what is done. I will explain this. This is what PCR, polymerase chain reaction. What is that polymerase chain reaction involved? Polymerase chain reaction enables us to get the gene of interest in multiple copies. Okay, so as I told you, what is this? It is a denaturation. For that, what is done? It is heated. The two strands of DNA, you can see that the two strands of DNA, it gets separated now. Okay, because of denaturation. I will come to that point, what is annealing. Now you can see primer here, primer. And this primer starts reading in this direction, and this primer starts in another direction. So this will make whatever this DNA we are interested, this is a region to be amplified, So this primer is required to start the process. So it makes, when it reads the frame, it makes that gene of interest to get amplified. And so whenever it passes to 30 cycle, it will be amplified by 1 billion. So let me explain this step, this polymerase chain reaction in 3 steps. First step is denaturation. Second step is annealing. Third step is extension of primers. We shall study that. Okay? This is what. The same thing, it is shown when it completes the 20 to 30 cycle from one copy is a 2 copy, 4 copy, 8 copy, like that goes on, and you will get millions and millions of copies. Okay? Let us see how it happens. Technique. The technique involves the following 3 steps. 1, denaturation of DNA fragment. The target DNA containing sequence to be amplified is heat denatured. That means it is heated around 94 degrees centigrade for 15 seconds. Always it is asked in the NEET exams and all. For how much time, at what degree it is heated? The target DNA means the target DNA containing sequence to be amplified is heated denatured to separate its complementary strands. This process is called melting of target DNA. Melting of target DNA. Okay. Target DNA contains sequence to be amplified is heated in nature. It is heated to the extent of 94 degrees centigrade for 15 second duration. When you heat this for 94 degrees centigrade for 15 seconds, the two strands get separated. To separate its complementary strand, this process is called melting of target DNA, or it is a denaturation of DNA fragment. Okay? Understood? Now, this is what you are doing denaturation. The two strands are together. When it is heated, you get the two strands to get separated. Annealing of primers. Primers are added in excess to the same. Primers are added in excess, and the temperature is lowered now. First, it was a high temperature of 94 degrees centigrade. The temperature is lowered now. it is lowered to about 68 degrees centigrade for 60 seconds. At the beginning, 15 seconds. Heating at 94 degrees centigrade for 15 seconds. Cooling at 68 degrees centigrade for 60 seconds. As a result, the primers form the hydrogen bond and anneal to the DNA on both sides of DNA sequence. So you have two strands separated. The primers, they get attached. The primers form hydrogen bonds and anneal. Annealing is actually attachment, holding tightly. Anneal to the DNA on both sides of DNA sequence. On both sides of DNA sequence. You can see here, this is what? Annealing. Okay? Annealing. Okay? So these are the two primers. Primers are nothing but a sequence, the oligonucleotides. A small oligonucleotide means not too long. It is a small with very few nitrogen bases. Okay? Alright. Next step you see. Primer extension. You have seen that primer? At a 2 point. The primer has to extend. Primer extension. Finally, different nucleoside triphosphate, DATP, DGTP, DCTP, DTTP, different nucleoside triphosphates, okay, and a thermostable DNA polymerase, what we call TARC polymerase. Why it is called a TARC polymerase? It is obtained from Thermus aquaticus. Thermus, TAQ, TARC polymerase from Thermus aquaticus and Vent polymerase from Thermococcus litoralis are added to the reaction mixture. When after the cooling, these are added to the reaction mixture. It helps in polymerization, making all these nitrogen bases and nucleotides to join. It helps in polymerization process of primers and therefore extends the primer at 68 degrees centigrade, resulting in the synthesis of multiple copies of target DNA. Multiple copies of target DNA sequence. So one primer is situated at one particular end, another gene of interest. We want that gene of interest. The gene of interest has two strands. So one primer here, it contains like this, another primer here, it contains like this. So this extends the extensive primer, resulting in synthesis of multiple copies of target DNA sequence. Okay? So first one is heat denaturation of DNA. Second one is annealing. Third step is primer extension. And this is what? You see now? It was here. It started now. Annealing. The primer extension. This is extension. So you see that the DNA gets extended. That means we wanted to get all these. These are all what we have, what we want. This particular gene of interest, we have to multiply. Okay? Now, after completion of all these three steps in one cycle, again the second cycle repeated following the same process. If 20 such cycles occur, then about 1 million copies of target DNA sequences are produced. Okay, recently this technology has been improved much more, where instead of TARC polymerase, the RTF, that means from bacteria Thermus thermophilus, Thermus thermophilus, so TH, RTF polymerase is used, which transcribe RNA to DNA and therefore amplify the DNA, which transcribe RNA to DNA. Okay, this is what RTF you have, RNA to DNA. So DNA, this is another method. RNA to DNA, there but amplify the DNA. Right? Okay, this is what I have seen. What is this procedure? First one, heat denaturation, 95 degrees centigrade. You see that this is what we have, gene of interest. So it gets separated, two strands get separated. And here you one primer. Here whatever complement is there, that base will come and you get from one DNA strand, you got now two gene of interest. So this will continue, this will continue. And this again, continue like that. So in this way, you do come across, within short time, you do come across billions of, in a 30 cycle of that, it comes, you can calculate, it comes out to billion. Right? Clear? Hello? Is it clear? Do you have any doubt? Any doubt? Anand
No, sir.
students? Anand students, boys? Harshvardhan? Sarvottam? Charan?
No, sir.
Charan? Who else is there? Arshivardhan, Sarvottam, Charan. Then who
Sir.
else? Madam? Siddesh?
Yes, sir. Understood, sir.
Is it clear? Understood?
Yes, sir.
Yes, sir.
Sir, Chinmay.
Namrata? Are you there?
Yes, sir.
Oh, understood?
Yes, sir. Understood.
Okay. Nadira? Visvita?
Yes, sir.
Understood?
Yes, sir.
Tanusri, Jayasri, Savitri,
Yes, sir. Yes, sir. Understood.
Yes, sir.
Kanisa, Aisha, Blessy, Navya,
Yes, sir.
Navya,
Yes, sir.
one more girl,
Yes, sir.
Rafa,
Janani.
Yes.
Janani. Who to? Janani, Kanisa?
Yes, sir.
Okay, your friend. Okay, shall we go ahead? Now, insertion of recombinant DNA into the host cell or organism. What you have to do? Insert this into the host cell. We got multiple copies of gene of interest. There are several methods of introducing the ligated DNA into recipient cells. Ligated DNA into recipient cells. How? Several methods of introducing the ligated DNA. Recipient cells, after making them competent to receive, take up DNA present in its surrounding. So, if a recombinant DNA bearing gene for resistance to an antibiotic, ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin-resistant cell. You know, by default, or E. coli, Escherichia coli, is without any resistance to antibiotic. It has no plasmid having any resistance to antibiotic. If at all you introduce recombinant DNA into E. coli, recombinant DNA through plasmid into E. coli develops ampicillin resistance. Okay? Right? Now, if you spread any transformed cells on agar plates, transformed cells are the cells which received foreign plasmid. If at all this plasmid has a recombinant DNA, we call them recombinant varieties. If you spread the transformed cells on agar plates containing epithelium, only transformants will grow, untransformed cells will die. Okay? Because E. coli now developed ampicillin resistance, which is a transformed. What is a transformed? A transformed cell is one to which we have introduced the plasmid with recombinant DNA. Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. Whatever that will survive, that is indication that they got ampicillin resistant gene. Whatever this E. coli die, it is indication that they did not possess ampicillin resistant gene. So the ampicillin resistance gene in this case is called selectable marker. Selectable marker, right? Where due to ampicillin resistant gene, one is able to select a transformed cell in the presence of ampicillin. So once due to ampicillin resistant gene, now if the ampicillin resistant gene is introduced into the E. coli, E. coli it was not having any plasmid with ampicillin resistant gene. Once this E. coli get this plasmid with ampicillin resistant gene, it can now survive when it is put in ampicillin. So ampicillin resistant gene in this case is called a selectable marker, because tomorrow you can add any of the gene of interest, you can ligate it into ampicillin resistant gene area. What is that? That area is marked by what? In which part of B322 you have ampicillin resistant gene? What is that mark area is called? It has which type of restriction enzymes? Hello?
PST ones.
PH1 and
PSI.
and
PVU1, sir.
Yeah, PV1. Okay, good. Right. So obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. You must know, I have already told you, whatever you do under recombinant DNA technology, ultimately what we are interested, in getting desirable protein. Hence, there is a need for the recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. So, recombinant DNA getting expressed means whatever recombinant DNA you are getting inserted, that recombinant DNA should show you the production of a desired protein. The foreign gene gets expressed under appropriate condition. The expression of foreign gene in the host cells involve understanding many technical details. After having cloned the gene of interest, that means you have multiplied. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing them on a large scale. We want to get to that recombinant DNA, whatever we have done, and we know that it is producing a desirable protein. We want to get to this in a large scale. If any protein encoding gene is expressed in a heterologous host, heterologous, not heterozygous, if any protein encoding gene, that is recombinant gene, if any protein encoding gene is expressed in a heterologous host is a host which expresses the protein in the cell lines where they are not supposed to be produced. That means different. It is called recombinant. It is called a recombinant protein. If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. It is not naturally produced by that particular organism, but it is produced by recombinant technique. Therefore, it is called recombinant protein. The cells harboring cloned genes of interest may be grown on a small scale in the laboratory. The cells harboring cloned genes of interest, that means bacterial cell into which we have introduced this recombinant DNA, or we have introduced it into gene of interest, may be grown on a small scale in laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. So whenever we have administered this recombinant DNA, or we have through recombinant DNA technology gene of interest, it brings the production of expected desirable protein. And after that, it has to be purified by different techniques. The cells can also be multiplied in a continuous culture system where the used medium is drained out from one side while fresh medium is added from the other side to maintain itself in their physiological most active log or exponential phase. This type of culturing method produces a larger biomass leading to higher yields of desired protein. What is done? The cells, whatever the cells are now getting this desirable protein produced, the cells can also be multiplied in a continuous culture system wherein the used medium is drained out. That means you are culturing that in one particular medium. You are getting that cells which are going to give you the desired protein. So you are culturing them in one medium. And what is done here in this medium, where used medium is drained, you have put in a big, let us say in a big vessel, you have put some medium. Medium is to enable that bacteria to feed and to grow. So from one side, a fresh medium is coming, from another side, used medium is going. Okay, so wherein the used medium is drained out from one side, while fresh medium is added from the other to maintain the cells in their physiological most active log or exponential phase. This type of culturing method produces a larger biomass leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of product. To produce in large quantities, the development of bioreactors, where large volumes, 100 to 1000 liters of culture, can be processed, was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc., using microbial plant, animal, or human cells. A bioreactor provides the optimal condition for achieving the desired product by providing optimal growth condition, temperature, pH, substrate, salt, vitamin, oxygen, etc. The most common use of bioreactors are of stirring type, which are shown in figure 11.7. See, this is what you have. Simple stirred tank bioreactor, and this is a sparged stirred tank bioreactor. Okay, these are the two. So no more details of this are given. You are simply given the two figures to showing you that there are two types of bioreactor. One is the simple one, and another is the sparged one. Okay, so this is, you must know what all different parts are there in these different types of bioreactor. So bioreactor is mainly meant for increasing the quantum of production of that protein, which we are getting through recombinant DNA technology. So proteins that are produced, they are purified and get converted into the desirable form. So this is what you have. The two types of the bioreactor, simple one. Okay. A stirred tank reactor is usually cylindrical or with a curved base to facilitate mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled to the reactor. We are not going to study the details of working of that particular thing. If you look at the figure closely, you see that the bioreactor has an agitator system. Agitator system is to just a mix, just like your mixer. Agitator system, an oxygen delivery system, and a foam control system. Okay, we can share the foam breaker, foam control system, that oxygen delivery system, and a temperature control system, pH control system. You see that this all that whatever labeling is there for that. Okay, pH system and sampling ports so that small volumes of the culture can be done periodically. Right? This is what you have. Now, the last method here is a downstream of processing. That means once you get a desirable the protein in a large scale, how to market it, how to use further. After completion of the biosynthetic stage, the product has to be subjected to a series of processes before it is ready for marketing. The processes include separation and purification, which are collectively referred to as the downstream of processing. It is asked for 4 marks. What is the downstream processing? The process include separation of the end product and purification, which are collectively referred to as the downstream processing. The product has to be formulated with the suitable preservatives. Some formulation had to undergo thorough clinical trials, as in the case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. So you must see that it is formulated with the suitable preservative, because a lot of money is getting spent. You cannot expect it to get spoiled easily. It should have a long life. Such a formulation has to undergo thorough clinical trial. If it is drug, it has to be clinically tried. It has to be tested in different animals, in different conditions. Strict quality control testing has to be for each product has to be maintained, and the downstream processing and quality control testing vary from product to product. Okay. Now, I would like to know: do you want anywhere any further details of this? Come on. Hello. Let us start. Kanisa, Aisha,
Yes,
Betsy,
sir.
any explanations required? Navya, anywhere? Janani, is it understood? I know
Yes,
that
sir.
this is little bit big topic, and yeah, come on, understood?
Yes, sir.
Anyone wants any explanation on any of this part right from beginning? So at this point you are not understood. Vismita,
Yes, sir.
Nadira,
Yes, sir.
shall we continue or you want any explanation? Jyoti, are you there? Jyoti, Prithvi, Namrata, Lohit,
Yes, sir.
is it clear?
Yes, sir.
Yes, sir.
Pardon? Hello? Okay. Savitri, Kavita, Tanusri, Jayasri.
Yes, sir. Yes, sir.
Shall I continue?
Yes, sir.
Okay. Now, you please make it a point. You please get prepared. We have to discuss some MCQs, am I right? On which topic? Hello. Which topic? Navya?
Human health and diseases.
Pardon?
Human health and diseases.
Human health and diseases we are not discussed. Hello?
It is done, sir.
MCQs? You don't know? Have we discussed the strategies? Are you looking into Google Classroom? Hello? Hello? How many of you are looking into Google Classroom? How many of you are looking into that? Jayasri, Sarvottam, Fatima, Rafa,
Yes, sir.
tell me which MCQs we have to discuss.
Human health strategies,
Human
evolution,
health. Yeah,
microbes.
three chapters are pending. Right? We shall do
Yes,
it.
sir.
We shall do it in tomorrow, day after tomorrow. Tomorrow is not possible. You please be ready with each topic, or you want me to put a solution key to that, and you'll ask the doubt. What do you have? Kenisa, what do you want?
In the class, we'll do, sir.
Dinner?
Class.
Class we shall do? Okay. We shall take up next class, if at all some special class, if at all I'm engaging on Saturday or Sunday, I shall take up human health and disease. Okay? Okay? And then come prepared for the next one, strategies in enhancement in food production. Okay, right. So now I shall take up the next topic, right? What is the next topic? Mike, yeah, next topic, biotechnology
And its application.
and its application. Are you seeing this slide?
Yes, sir.
Yes, sir.
Yeah. You know that in our day-to-day life, we are extensively using the result of biotechnology and its products. So, biotechnology, as we have learned from the previous chapter, that biotechnology process and principle essentially deals with industrial scale production of biopharmaceuticals and biologists using genetically modified microbes, fungi, plants, and animals. So we are using for our benefit. It is made in an industrial scale production. So biotechnology deals with the industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes or fungi, plants, and animals. The applications of biotechnology includes therapeutics. What is that? Treatment, therapy, or drug. One word for this: treatment of the diseases, or what you say, therapy or drug. It is together called therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. All these are the direct effect of applications of biotechnology. Okay. Now let us look into these one by one. Three critical research areas of biotechnology are, three areas are very important. One, providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Two, creating optimal conditions through engineering for a catalyst to act. Three, downstream processing technology to purify the protein or organic compound. These three are very important research areas in the field of biotechnology. Providing best catalyst in the form of improved organism or a general microbe or providing pure enzyme, creating optimal condition through engineering for a catalyst to act, and downstream processing of technology to purify the end products of protein or organic compound. Let us now learn how we have used biotechnology to improve the quality of human life, especially in the field of food production and health. Okay, let us take up this. Biotechnological application in agriculture. Let us look at the three options that can be thought for increasing food production. How we can improve the food production? One, agrochemical based agriculture. Agrochemical based agriculture. Agriculture where extensively chemicals are used that enables us to get the product more. Organic agriculture. And third one, genetically engineered crop based agriculture. We shall see that which crops are genetically engineered. So these are the three which have resulted increase in the food production. One, usage of the chemical in the agriculture, organic agriculture, and genetically engineered agriculture. So, the green revolution, what do you mean by the green revolution? What is the green revolution? Anyone? Anyone? You have heard about that? What is green revolution? Rakesh? How that green revolution has resulted in increasing the food production? Hello? Rakesh, are you there? Rakesh?
Yes, sir.
Can you tell me how that, what is the green revolution deals with? What are the main parameters in green revolution? Anyone else? How that through green revolution we have increased the food production? Anyone? Kanisa? Namrata?
Yes, sir.
Namrata? Yeah, come on, who will tell? Kanisa, tell me. Kanisa, tell me. You know? Do you know?
Don't know, sir.
Green revolution has enabled us to, or by green revolution, we have learned that we have learned the use of chemical fertilizer, inorganic fertilizer. We have learned the way of going for hybrid varieties of crops. We have learned to use insecticide and pesticide. All these were not there prior to green revolution. Prior to green revolution, local varieties of crops were grown. Organic manuring was done. Very, very less insecticides or pesticides were used at that time. The green revolution succeeded in tripling the food supply, but it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties. That's what I said. But mainly due to the use of better management practices and use of agrochemicals, fertilizers, and pesticides. And these have enabled us to improve the food supply, improve the yield, crop yield. However, for farmers in the developing world, agrochemicals are often too expensive. Spending a lot of money on these fertilizers and pesticides. and getting very less yield, it will be too expensive, and further increases in yield with existing varieties are not possible using conventional breeding. If it only go for conventional breeding type, good old methods, it is not possible to increase the yield. So what has to be done? What has to be planned? Is there any alternative path that our understanding of genetics can show that farmers may obtain maximum yield from their fields? Forget about using more and more inorganic fertilizer. Forget about using better crop varieties. Forget about using pesticides and more fertilizers. Is there any better way of getting better yield by understanding of genetics? Is there a way to minimize use of fertilizer? The need of the day, the need of the hour is to minimize use of fertilizer and chemicals so that their harmful effects on the environment are reduced. Now, if at all I ask the question to you, you are using these inorganic fertilizers to get a better yield in your cropland. Is it all these inorganic fertilizers are 100% used by these crops that you are growing? Ms. Smita, whenever we apply inorganic fertilizer, is it that all these inorganic fertilizers, whatever you put, 100% are utilized by the crop that you are growing?
No, sir.
No. What will happen to that extra fertilizer that you're putting? What will happen to that insecticide or pesticide you're putting? Don't you think that this will cause environmental pollution? So, use of genetically modified crop is a possible solution to minimize this effect of fertilizers and other chemicals on the environment. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. You will be asked what is a GMO? Plants, bacteria, fungi, and animals whose genes have been altered, of course, by technology, recombinant DNA technology, are called genetically modified organisms. GM, genetically modified plants, have been useful in many ways. Genetic modification has made crops more tolerant to abiotic stresses like a cold, drought, salt, heat. You do get the crops which do show generally our crops are getting affected by sometimes extreme cold in winter or drought in summer, salt many times, or by heat. And it is possible to get the crops of better variety which have the resistance to abiotic stresses. Okay? Or reduce the reliance on chemical pesticides. We have to find out the way without using pesticide or with less usage of pesticide, how can we protect our crops? Reduce the reliance on chemical pesticides, helped to reduce the post-harvest losses. Many times, once these crops are harvested, before taking them for marketing, they get spoiled. Now, let me just tell you, if at all you are growing this tomato, okay, let us say that when you are getting your crop in a certain tons, if at that time there is no rate at all, if you don't want to buy that in the market, you cannot buy. Such a low rate has come because every farmer has started growing this tomato. Demand is less, production is more. If at all you think of some technology, biotechnology application, and see that this tomato is somehow it is stored and it could be used in a different way and marketed in a different way, don't you think that it will be great help? So this is what? Biotechnology and application help to reduce the post-harvest losses, increase the efficiency of a mineral usage by plants, increase the efficiency of mineral usage of plants. This prevents early exhaustion for fertility of soil. Now, what is happening? Because of usage of fertilizer day in and day out, repeated use of inorganic fertilizer every year for every crop, many times in a year, what has happened? The fertility of the soil is getting lost. Unless you put this inorganic fertilizer in that particular soil, you cannot grow anything because there is no rejuvenation of the soil. Soil is providing only and support to the plant and nothing. Nutrients you have to provide. It is only providing anchorage to the plants. Enhance the nutrition value for food. Example, vitamin E enriched in rice. Whatever that crops you are growing, if it are that the crops, that are whatever the vegetables, whatever fruits, whatever food grains you have grown, if it are, they are found to be more and more tasteful, these are all applications of biotechnology in different fields. In addition to these uses, genetically modified crops has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, pharmaceuticals, etc. I hope this is what the introduction part with regard to the biotechnology and its application. So we are genetic modification has all these implications. Understood? This will be asked for 3 to 4 months question board examination. Some of the applications of biotechnology in agriculture that you will study in detail are the production of a pest-resistant plant. What is pest? Pest is unwanted guest. So we should go for pest-resistant plants which could decrease the amount of pesticide use. Today, what is most important is that in order to get to more and more yield, extensively, the inorganic fertilizers are used and also to control this pest, insecticide, pesticide, herbicide, all these are used. All these both extensive use of inorganic fertilizer and extensive use of these pesticides and all, these are going to pollute our environment more and more. So we must think about to reduce both of these. Both of these. So that is what we are going to study in this particular chapter. Bt toxin is produced by a bacterium called Bacillus thuringiensis. I shall start from here, Bt toxin, and I will continue in my next class in the next week. Okay? Right?